Good afternoon. It's my pleasure to be here, and I hope everyone has their translators going. Yes? See? Okay, so today I'm going to tell you about work that we are currently doing on pecan genetics and about clonal pecan rootstock. So currently there is an effort at creating genetic tools for pecan. And this effort is being funded by the United States Department of Agriculture. And it is composed of a number of groups, including New Mexico State University, USDA, including USDA, the Carrier Germplasm Repository in Texas, USDA location in Georgia, another USDA location in Louisiana, the Nogal Foundation in Oklahoma, the University of Georgia, and the University of Arizona. And together we have composed a team of pecan researchers who are striving to look into the genetics, and we're running at a very fast pace. So it's the genome era. So a genome consists of all of the genes and the genetics in an organism. How many of you have had your genome sequenced? Anybody? For a few $100s, you can have your genome sequenced, and you can find out where you're from, or you can find out if you might be predisposed to some genetic diseases. Well, today we're working on pecan. So, if you've had your genome sequenced, can you tell me how many pairs of chromosomes humans have? Richard, let's see if you can get it right today. And he said 23 pairs, right? Okay, well, pecans have 16 pairs of chromosomes. And this is just to remind you how pecan breeding works, so to speak. So just as in humans, pecans have a male and female, and each of the progeny gets half of the genes from dad and half from mom. The DNA is recombined in each part in order for us to get our gametes. And because of this, we get genetic diversity. And this genetic diversity is very important. So as we were told previously, pecan is native to North America, and the native region extends up into the United States, far north, all the way down here. We have distinct populations here in Mexico. And these populations, these native pecan populations here in Mexico, are very genetically distinct from other wild native populations in the United States. And it's because of the genetic diversity that is within these trees that allows the trees to grow and grow very well in these different environmental regions. So for instance, we have very hot regions, well, as you know, here in Mexico, and then we have some colder regions up north. Just so you know, Georgia and New Mexico, and we produce pecans, not as many as you guys have been producing lately, but we produce quite a bit of pecans, and these are not the native regions. So pecan has been planted in places where it is not native, and this is occurring worldwide. So here are some pictures of some native pecan trees, and native pecan trees can grow and they can thrive and live for hundreds of years. So genetics influence pecan trees in several ways. One, it influences tree growth. It can confer either cold tolerance or heat tolerance. It also controls the time of flowering and production. Also, nut production and quality is influenced. Water requirements, nutrient uptake, and scion and rootstock compatibility. All of these things are influenced by the genetics in the pecan tree. So this is just to give you an example. I am sure that all of you are very familiar with both Western and Wichita varieties. So I just put some differences here so we can compare them. So as you know, Western, as a variety, is a vigorous tree, and it grows very quickly, whereas Wichita grows slower. Western typically is a heavy producer. Wichita has larger nuts. Western responds well to pruning, and is less affected by water and heat stress, whereas Wichita is very susceptible to water and heat stress. Western is less susceptible to zinc deficiency, and Wichita has a major susceptibility to zinc deficiency. So all of these differences between Western and Wichita that you've already noticed are due to genetics. So work that is being done in pecan, there are several questions that we want to answer. First, we want to identify genes in the genome that are responsible for specific traits. And we also want to be able to genetically identify cultivars and varieties, so DNA fingerprinting. This is an example of a DNA fingerprint for pecan. I'm not sure that you can actually see these peaks, but if you can see these blue peaks, we can use this to tell the difference between Wichita and Western. And we can do this for a number of trees, but we want to be able to do it for almost every variety of cultivar out there. And so that is one of our goals. So for the last 100 years, when pecan trees are bred, breeders typically choose two good trees. They like the traits that they see in trees, and they decide to cross them. And then after crossing, they wait. We wait, and we wait, and we wait. And 20 years later, we decide whether that was a good cross. It takes a long time to bring out new varieties of pecan trees. And because of this, the genetic tools that we're building will decrease the time. So what we want to do, and what we are doing right now, is we are looking at the different cultivars, native trees, different varieties, and we are genetically sequencing. We are looking at their genotype, just like if you had sent in a DNA sample of yourself to get your genome. And then we are also looking at phenotypic descriptors. So if there is something specific about a tree, then we want to see what genes relate to that. For instance, in Georgia, scab is a major problem, major disease problem. It's caused by a fungus. And there are some trees that seem to display resistance to scab. So that's one phenotype. Also, we want to look at trees, we want to look at the architecture of trees. We'd like to see if we can maybe eventually have shorter pecan trees. We'd like to have trees that are resistant to Phymatoptricum, or PO. You might know it as cotton root rot. That's a problem out west and here in Mexico as well. Anyway, so we are looking at genetic sequences and linking them to phenotypes. And what we will be able to do is do what's called molecular assisted selection. And with this, we will be able to have better tools to where we can breed trees for specific environments much faster. So again, some of the genetic markers that we are looking at include specifically tree architecture for size control, nutritional acquisition, meaning some trees take up nutrients differently than other trees, and we'd like to understand why, disease resistance, and salinity tolerance. So some of the tools, exactly what we're doing is that we are looking for systems to identify specific pecan genotypes. So if you own a nursery and you need to check to make sure that you have the right genotype, you'll be able to have the tools to do that. Also, by October of 2017, we're going to have a website that's online, and new information about these tools and the research that's going on will be available to you. And then we are looking for markers for our faster breeding programs. So here again, I have a map of the native region for pecan, and you see these large red circles. We are completely sequencing we're doing complete sequences on 4 pecan trees, and the red circles indicate approximately where those trees came from, where their origin was from. So one of the trees that we have almost completed sequencing, we refer to it as 87 MEX 3.2.1, and this tree, seed was collected from this tree here in Mexico by L.J. Graupe in 1986. And he took seed back to Texas as an agreement, and he planted them. And this tree is the first pecan genome that has been sequenced. It is almost complete, and right now we're doing mapping so we can figure out what genes are on what chromosomes. We are also looking at Elliott, which is another cultivar variety that has some resistance to scab. We're also looking at Lakota and Pawnee. So those 4 pecan trees, those 4 varieties will be completely sequenced, and the data will be available. So if you're interested in looking at some of the pecan data or some of the genome annotation, meaning what genes are there, you can go to this website. It is, well, I don't see the HTTP, but if you search through New Mexico State for pecan genome database, you will find it. And here you can find the draft genome. This isn't the complete genome, but the complete genome will be up soon. So along with sequencing, having 4 complete sequences, we've also been working on a genome-wide association study. And this project has been done by collaboration with the University of Tokyo and the USDA and New Mexico State University. And what we've done is we have gone and we have collected we started with 1,000 genetically different trees. And in the end, we have looked at 800 different pecan cultivars. And we have sequenced them, not as a complete sequence, but with enough coverage that we can look at differences. And we can start correlating some phenotypes with some of the genetic sequence and looking to see where they were from. So we're actually correlating genotypes with the geographical place of origin. And these 800 different cultivars include seedlings from North America and from Mexico. And this project will allow for development of marker-assisted breeding. So right here, this is kind of like a phylogenetic tree. It's a tree as you will. And it's kind of like if you do your own genealogy. So if you have a family tree where you can see your grandparents and your cousins and so on, this is what this is in the pecan world. And as we've sequenced, this is just a subset of part of what we've sequenced. But you'll see that all of the trees that originated in Mexico, they group together, and they're closely related to trees that have originated in the southern United States. And the trees that come from the northern United States are distinct and separate differently from these. So what this tells us is that our data is good, our sequencing data is good, and it will be useful. And we can already see how genotype and geography have correlated. So with this, what we want to do is we're going to be able to find our DNA markers to track traits. So for instance, when pecan breeders breed trees and then they plant all of the seed and then they wait and they wait and they wait to see which tree has the traits they want, we'll be able to speed that time up. So instead of waiting for the trees to have flowers, what we'll be able to do is take the seedlings and check the DNA of the seedlings to see if it appears, if the markers that we're interested in are in those seedlings. And if they are, then we'll continue with those seedlings. And it will make screening for traits much more efficient. So we're going to switch gears a little bit, and I'm going to talk about the importance of rootstocks. So as you know, in all commercial orchards, so we're not talking about native trees now, but we're talking about trees that you typically plant for commercial use now. They are composite trees. They are composed of a rootstock at the bottom and a scion, which is a known variety, such as Wichita, Western, Pawnee. And the tops, if it's a Wichita, all Wichitas are the same. If it's a Western, all the Westerns are genetically the same. But the rootstocks are grown a little differently. We typically know who the mom of the rootstock tree was, but not usually the dad. So why do we care about rootstocks? Someone told me a few years ago that rootstocks were only for anchorage of trees. And although anchorage is really important, rootstocks play a critical role in pecan trees. The rootstocks actually influence nut quality, production, and disease resistance. The roots allow the tree to grow in diverse environments, whether it's alkaline or salinity soils. It can help control tree architecture. So, for instance, I'm interested in size control. And also, the rootstocks are also very important for nutrient uptake. Iron and zinc. And also for disease resistance. So the rootstocks play a critical role in pecan tree production. So what I want to do here is show you where we are today and where I anticipate we'll be in the future. And I'll show you why I think this. Okay, so today, all pecan rootstocks are grown from seeds. So nurseries plant seeds, so if you have Riverside or Burkett, whatever rootstock variety you have, those are all grown from seeds. So the paternal parent is unknown. Since pecan is wind pollinated, you know who mom is, but not dad. And so because of this, there's high genetic diversity with those rootstocks. So you may not know this, but as pecan nurseries grow the rootstocks, they grow the seeds, they go through and they separate the ones that aren't growing the same. But this difference in genetic diversity can lead to variability in orchards and orchard management. So in the future, what we're going to have, you're going to be able to have designer trees with desired traits for the region and for scion. So depending upon your geographical area, you'll be able to select trees that are specifically designed for that area. In the future, you'll know who both of the parents are, or you'll have the ability to know who both of the parents are. And clonal rootstocks will eliminate the genetic variability. But it's important to have complete analysis of clonal rootstocks. So what are clonal rootstocks? First, a clone. The word clone usually scares people. But in the pecan industry, you've actually been cloning for a very long time. You just may not have thought of it that way. So when you take graft wood and you bud it to your rootstock, that's cloning because, for instance, in your orchard, all the western trees are the same. So cloning is genetically identical individual produced by vegetative means. But if you've ever tried, it's very difficult to root pecans. If you take a cutting of pecans, it's not impossible, but it's very, very difficult. And so I'm going to talk to you about the use of micropropagation to using tissue culture to vegetatively propagate plants. Has anyone in this room seen plant tissue culture? Two people. Okay, thank you. Okay, so micropropagation. You can produce a large number of genetically identical plants in a very short time. So that's a real benefit. However, it's very labor intensive. Using micropropagation, you have the ability to produce plants that are clean of plant diseases, such as viruses or bacteria. And using micropropagation, you can effectively screen plants for specific traits. And I'll show you a couple of examples, such as looking at salinity tolerance. So first, the first stage is, I'm going to take you through micropropagation. So the first thing you do is, does anyone have a favorite pecan tree in your orchard? Anybody? Does anyone have a, okay, great. Someone back there has the best pecan tree in the world. What if you wanted 1,000 of them? Or maybe 1 million of them? So what you can do is you can start from a number of sources. You can start from your favorite pecan tree. You can take material from that, or you can start with seedlings and take material from that. And what you're going to do is you're going to surface sterilize it and introduce it onto plant media. This plant media is designed, it has all of the nutrients and the salts and hormones that pecan trees require. And because of that, they're able to grow and produce. So first we introduce into tissue culture. And next, we let them grow and multiply. So these are pecan trees in tissue culture. And those are multiple clones. So I can take a little piece of a pecan tree and make thousands of trees from that. And all of those thousands of trees will be genetically identical. Genetically the same. Just more cups. So I like to tease people that I think I have this largest, smallest pecan orchard. So I have about 100,000 trees in my laboratory in cups and different genotypes. Here's a picture of what the trees look like as they're growing. So believe it or not, each of these trees can potentially become a large pecan tree. And in one cup, sometimes we can have 50 trees So sometimes I say it's an acre in a cup. So after we produce trees, we then change how they're developing so they will produce roots in vitro conditions. And so all this picture is showing you are actual pecan roots in that plant media I showed you. It's so much better if you can see it in person. So if you're ever in New Mexico, feel free to call me or to email, and I'd be happy to show you some of these pecan trees. And here again, I'm just trying to show some of the pecan roots with the trees. So they're tiny little plants. Once they have roots, we then change and we plant them into soil. So we have to acclimate them into soil. And believe it or not, this is really a difficult part because they've had everything they've ever wanted for their whole little lives, and now they have to start working. It's kind of like teenagers as they approach adulthood and go off to college. So they acclimate, and we harden trees in the greenhouse. Here's an example of clonal plant roots 5 months after planting in soil. And just to give you an idea of growth, this picture right here was taken in March. And this is my greenhouse at New Mexico State University. And all of the plants you see are clonal pecans. They've all gone through micropropagation. And at this point, I was so proud of my trees. I thought, oh, they look so good. But this picture was taken in July. Can you see a difference in their growth? They've grown a lot in a few months. And here are the trees. And they're ready to go out into a field to be field tested, which is important. So cloning is here. And clonal pecan rootstock will become available. But I want you to really hear this, that it's the genetics of the rootstock that make the difference. You can have a good pecan clone and you can have a bad pecan clone. So it is very, 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 very important that if you are buying rootstock trees that are clonal, that they've been tested and that you know what you're getting. So very important. So this is the next step, field testing, to see how they'll work in the field. And you know, clonal rootstocks is new for pecan, but it's not new to other tree species, such as pistachio or almond, even walnut, there's walnut rootstock, apple, clonal rootstock. So now pecan is entering into the clonal age. So as a scientist though, as someone who loves genetics, this tool gives us the chance to study pecan trees in a way we've never been able to study pecan trees before. So typically with rootstocks, as I explained, they are planted by individual seeds. And each of those seeds are genetically different. And so that gives a lot of variability when you want to test rootstocks in the past. But now, we can now clone specific rootstocks, and we can have 50 or 100 trees that are genetically the same that we can test for certain stresses or for specific things. So I'm going to give two examples. One is salinity tolerance, and the other one is nutrient acquisition. So salinity, saline source, due to the types of water in the soil, having trees that are salinity tolerant are becoming more and more important. And so one of the traits that I'm looking at with the genotypes that I've cloned, and we've cloned a little over 300 genotypes, is now screening through all of those genotypes for salinity tolerance. And I have to tell you that out of 300 genotypes that we have cloned and that we have screened, only 12 of those genotypes have shown real salinity tolerance in a lab condition. So the first thing we do is we take, this is again our plant media, and now we've introduced salts. And here I'm just showing you sodium chloride. We've done some other salts as well now. And we put our genotypes onto a base amount. If the plant dies, as you can see, this plant is really brown. It did not survive the salinity testing. It's not tested any further. But if it does survive and does well, then we put it onto higher and higher levels of salt to see how well it can do. And then these are then planted. And for each of the genotypes we've done, we have tested somewhere in 30 to 50 shoots per genotype per salinity test. That's a lot of trees to test. But because of this, we have good statistical power, and we really can say without a doubt that these trees in laboratory conditions had salinity tolerance. So here's some more pictures of some other genotypes, our controls, and then you can see on differing amounts how some of the trees did. This tree didn't do so well on salinity, on high salinity tolerance. It didn't have salinity tolerance, whereas some of the others did. So the trees that have shown high salinity tolerance have been planted, and they'll go into orchards for testing in a field trial. Okay, so all of us saw Jim's talk a little bit ago. Can anyone tell me what this tree is lacking? This is a clonal pecan tree. Can you guess what this tree needs? Anybody? Nickel. Good. And it's right there in the title. So, this tree is deficient of nickel, and actually we had quite a few trees that were deficient of nickel. And so, in a study that we've been working on, we took 2 genotypes, so 2 different genotypes, and we had 30 trees in each trial. And I'm not going to show you the whole trial today. But we gave them different amounts of foliar nickel. And so this tree was treated with nickel, and with 1 week, look at the difference. So this was at 250 parts per million. But within 1 week, we would see astonishing differences. So with our 2 genotypes, we tried a number of things. First off, we tried different nickel concentrations, and I don't have the graphs for all of them here on the slide. But we tried everywhere from 100 part per million all the way to 1,000 part per million. My goal was to see what nickel toxicity looked like. And I believe we actually got to it. We have some trees that aren't doing so well. With my clonal potted trees, so I have to say that, this is different than a field environment because they are in a greenhouse. But you can see that if I showed you all of the graphs, you would find that anywhere from 100 part per million to 400 part per million on the clonal trees worked just fine. And in fact, 250 looked great. But once we exceeded 800 part per million and 1,000, we actually had detrimental growth on the pecans. When we looked at the 2 genotypes, it's very interesting because one of the genotypes is a Western origin. I'm not sure who that is yet. I haven't tested him. And one's a Wichita. And what's interesting is that the Wichita tree seemed to respond better to the nickel. We saw better growth and more leaf development. And we also have data to show that there are differences in how these two genotypes, they've been given the same amount of fertilizer, of zinc, of nickel, and yet they're accumulating the nutrients differently. The way they act, the way they're taking up the nutrients, I don't know if that's different, but there is a distinct difference between the genetics in these two trees and how they are taking up nutrients. So if you were to buy pecan trees and you knew ahead of time exactly what the pecan tree needed, that would be a benefit to you, instead of having to do guesswork like what we're doing and studying right here. So there's a lot more to this study, and I'll just show that to you another time. But what I wanted to show you is the power that these clonal trees have in research, not just in the real world, but they will be available within the next 5 years. And so it will change the way pecan orchards are planted in the future. Okay, well with that, the slide's a little dark, but I wanted to thank some people or acknowledge some people who've done some research. In my lab, I have quite a few. Unfortunately, I can't even read their names. But my graduate student, Kimberly Cervantes, tried to put some of the slides in Spanish and Espanol for you. And I also work with Dr. Richard Karama, and then my colleagues from USDA in Texas, LJ, University of Tokyo, where we're doing the genome-wide association study, and Hudson Alpha, who is completing the sequences of the 4 trees, and funding agencies. We've been funded by private growers, AUP growers, along with the USDA and New Mexico Department of Ag and Specialty Crops. So with that, I would be happy to answer any questions.
¿Tienen alguna pregunta? Un aplauso primero. ¿Pueden encender todas las luces, por favor? ¿Tienen alguna pregunta para la doctora Randa? Allá está una pregunta. Un momentito. ¿Tiene el micrófono por allá? Un momento. Ahí va.
Hola, buenos días. Buenas tardes. Doctora, ¿cómo pudo establecer en el laboratorio a partir de brotes en los árboles? ¿O cómo se lo llevó al laboratorio los explantes? Vi que tomó una semilla en un árbol, la germinó en cajas Petri y de ahí tomó los explantes.
So you can put either parts of a mature tree or seedlings into tissue culture. So you can do either way.
Dice que se puede tomar parte de la planta tanto de adultos como de las plantas de árboles adultos como de plantas de cualquiera de los dos.
No fue muy difícil traer el material de campo. No hay, muchos problemas con oxidación, contaminación, o nada más le dieron un tratamiento especial.
So if you are taking material from a mature tree, from an orchard to your laboratory, or my laboratory, you need to do it very quickly. You want to take the sample and keep it cool, if that's what you're asking. Yes? ¿Sí?
Sí, está bien. Solo tengo una pregunta más. Las plantas que se están llevando a las plantilitas ya al campo a hacer pruebas, ¿están respondiendo bien? ¿No están batallando que, no sé, tenga que hacer mayor adaptabilidad en malla sombra o que tengan buen desarrollo al ser plantas traídas del laboratorio?
So I don't know. My trees are ready to go to the field, and they were going to go the beginning of August. But I was waiting for the irrigation system to be ready, and it just is now ready. And to be honest, I'm scared to move them. I'm not sure there's enough time for them to be planted and, acclimate to the field. And then we have to turn them off before the freeze. So I think they'll be planted in the spring. So I don't know how difficult it will be from going from the pots to the field.
Perfecto, gracias.
Los árboles con resistencia o genes resistentes a salinidad, ¿cómo esperan que respondan en una condición normal de baja salinidad? How do you expect from those trees tolerant to salinity, how do you expect they will grow in a good condition?
They'll grow well. They'll grow without the salinity stress.
They will,
Yes.
well, the idea is normally when you have a salt resistance, a salt tolerant trait, they will spend more energy in order to do that. They need certain metabolites in order to have the salinity resistance. So if you place it in a very good condition versus another tree which doesn't have salinity resistance, they will be in lower condition for growing. That's what they will say, they will expect.
I showed you my trees, but what you didn't see is how long I thought they took to grow, so maybe I didn't recognize that.
¿Otra pregunta? Bueno, yo tengo una pregunta. ¿Han identificado materiales genéticos que pudieran ser enanizantes?
So we haven't found dwarf trees yet, but what we're hoping is that one of our breeding populations that we're looking at right now, we've crossed a Mexican variety with a Lakota tree. And because of the difference in architecture between the two, we think that we're hopefully going to get some resolution to looking at tree architecture, and from there, looking at some size control. That's my goal. I want small pecan trees someday.
Sí, le pregunté que si había algunos candidatos para tener insectos enanizantes, ¿no? Y me dice que no, pero que hay algunos que tienen una diferente arquitectura y que en un momento dado pueden ser candidatos a tener árboles con menor porte, ¿verdad? ¿Alguna otra pregunta? Bueno, doctor Vicente Lee, a ver un momentito.
En español, cuando obtiene la planta del cultivo de tejidos. Yo tengo un árbol muy bonito. I have a pretty nice tree. And I keep branches. And then I put it on this orchard. What? I get my tree
Yes.
from the sugar. I planted that tree direct to the soil. I need to use a rock stock for that one. Le pregunté que si cuando yo tengo mi árbol muy bonito, el que más quiero en toda mi fregada huerta, ahí tomo ramas, tomo yemas, los pongo en el cultivo de tejidos. Ahí obtengo mi arbolito. Ese arbolito lo planto directo en el suelo y va a ser igualito al árbol que yo tomé, o lo injerto sobre algún patrón. You get my question?
No, sir. Someone translate for me?
Well, I'm going to try again. I use my tree, my very nice tree.
Yes.
I took some pictures of that tree and put it in a tissue virtual,
Oh,
okay?
okay, so we want to clone your tree.
And then I
Yes?
come out with my small tree.
Okay, yes. Lots of trees. We're going to clone lots of trees, right?
Okay, I put it.
Okay, thousands.
One tree. I did plant that tree directly in the soil, or I need to use a root stock to put that tree. Que si el arbolito se lo planta directo, o lo invierto en algún
Are you wanting the scion or are you wanting the rootstock of the tree? Wait, what part?
I want exactly the nice tree that I have.
Is it a native tree or is it a tree that has a rootstock and a scion?
No, that's a tree, normal tree.
Well, so it depends on what your tree is. If it's a native tree, then we can clone that. But if it's a tree that's in a commercial orchard where it has a rootstock and then a scion has been grafted, it depends on what part of the tree that you're wanting.
He said that the tree that he could create when we planted.
Yes. Yes. Yes.
Dice que no necesitan propagar. Dice que si lo vas a plantar directamente, por ejemplo, si agarras una ramita de cuchita, es el cuchita que ese, ¿no? Ese tú lo agarras y lo propagas. Ya se propagó, vas y lo plantas, va a ser cuchita. Exactamente.
Exactly.
Bueno, le vamos a dar gracias